The Tasmanian tiger, easily distinguished by his straight, unjointed tail, is also a dangerous opponent, though, like the devil, is now very rare, being forced out of its natural habitat by the march of civilization.
Deep within the dense, wet eucalyptus forests of northwestern Tasmania, a remote field research camera triggers in the absolute stillness of midnight. For nearly 9 decades, scientific consensus has held that this landscape is entirely devoid of its apex marsupial predator. Yet, the raw, unedited night vision footage reveals a silhouette that defies the history books, an animal exhibiting structural traits that should no longer exist on this planet. Is it a miraculous remnant of a species that evaded eradication? Or are the shadows playing a cruel trick on modern zoology? Let us examine the physical evidence left behind on the lens. Zooming in to the terrifying footage. So, to evaluate the validity of any newly surfaced field footage claiming to document a living thylacine, Thylacynus cynocephalus, one must bypass emotional reactions and subject the subject's morphology to strict anatomical scrutiny. For nearly a century, our understanding of this apex marsupial predator has been restricted to dried museum skins, fossilized skeletal remains, and fewer than a dozen short sequences of black and white film recorded in captivity prior to 1936. When a remote trail camera captures a clear profile of an unidentified quadruped in the Tasmanian bush, field zoologists look for a highly specific suite of skeletal and behavioral diagnostic markers. These physical traits are unique to the family Thylacinidae and are entirely absent in the placental canids, such as feral dogs and foxes, that currently inhabit the Australian landscape. The most dramatic diagnostic marker occurs when the animal displays defensive behavior or executes a threat yawn toward the camera lens. In the recorded sequence, the creature's lower jaw drops until the maxilla and mandible form an astonishingly wide angle. Historical accounts from early European naturalists frequently asserted that a startled thylacine could open its mouth to a vertical 120-degree gape, a feature often cited as evidence of a semi-unhinged jaw structure resembling a reptile or a saber-toothed cat. Modern veterinary craniomaxillary analysis clarifies this phenomenon. While a true 120-degree vertical opening would compromise the structural integrity of the temporomandibular joint, the thylacine does possess an extraordinary, documented gape reaching up to 80 or 90 degrees. This extreme vertical clearance is made possible by a highly specialized, elongated articular process at the rear of the jaw and a unique alignment of the masticatory muscles. When the animal in the footage lowers its jaw to this extreme angle, it reveals an elongated, narrow muzzle containing a specialized dentition of 46 teeth, distinct from the 42 teeth found in placental dogs. The sheer vertical expansion of the maw creates a hollow, cavernous profile that cannot be replicated by any living canine, providing a definitive osteological anchor for species identification. As the animal shifts its position to navigate the dense undergrowth of native ferns and wet eucalyptus trees, the camera captures a clear lateral view of its torso, exposing the unmistakable transverse lumbar striping pattern. On a living specimen, these markings are not mere surface pigmentations. They are highly defined, dark brown to black bars that radiate outward from the dorsal midline across the hindquarters. A precise count reveals between 15 and 20 distinct stripes, which are concentrated heavily over the rump and the base of the thighs, completely fading out before reaching the shoulders or the forelimbs. In a pristine daylight context, these stripes provide critical disruptive camouflage within the dappled, shifting light of an old-growth rainforest canopy. On a low-light or infrared field recording, the high contrast between the dark bars and the surrounding sandy, yellowish-brown guard hairs creates a striking visual pattern. To confirm a thylacine profile, researchers look at the spacing and taper of these bars. They must remain perfectly parallel, growing progressively shorter as they approach the mid-back, rather than swirling or blending like the coat patterns found in domestic canine breeds or feral striped hyenas. Moving lower down the anatomy, The transition from the pelvis to the tail provides perhaps the most ironclad structural evidence against a canine misidentification. A domestic dog or a wild dingo possesses a highly flexible, expressive tail that attaches to the posterior pelvis via a narrow, distinct joint, allowing for free lateral and vertical movement. The tail of the animal in the new footage exhibits a radically different marsupial morphology. It features an incredibly thick, robust, and compressed base that emerges seamlessly from the lower lumbar vertebrae and gluteal muscle mass. This creates a rigid, tapered extension of the entire spinal column, structurally analogous to the heavy tail base of a kangaroo or a Tasmanian devil. The proximal half of the tail lacks any significant lateral flexibility. It cannot be wagged, curled, or hoisted over the back. Instead, as the creature slinks through the brush, the heavy rudder trails stiffly behind the torso, dropping toward the forest floor in a straight, unyielding line. When the animal alters its direction rapidly, the tail acts as a counterweight, pivoting stiffly from the pelvis to stabilize the center of mass during sudden lateral movements. Finally, the definitive test of the thylacine's presence lies in its biomechanical locomotion, which is entirely distinct from the cursorial gait of placental pack predators. When moving at a leisurely pace, the animal exhibits a slow, stiff-legged, and somewhat uncoordinated walk, planting its feet in a plantigrade or semi-plantigrade manner that suggests a reliance on deliberate stealth rather than long-distance endurance. However, the true revelation occurs when the animal is startled by the camera's infrared trigger and breaks into its high-speed escape gait. It completely abandons a standard mammalian trot. Instead, it shifts its weight backward onto its elongated hind limbs and executes a series of frantic, top-heavy, semi-bipedal bounds. This awkward, hopping motion, frequently detailed in 19th-century bush journals but rarely captured on film, is a behavioral artifact of the animal's specialized pelvic structure and reduced clavicles. The creature propels itself forward with rhythmic, bounding leaps, using its stiff tail as a spring-like stabilizer to maintain balance upon landing. This clunky yet remarkably rapid locomotion style is anatomically impossible for a wild dog or fox to sustain. When a field recording combines this unique bipedal bounding with a rigid marsupial tail base, a highly defined posterior striping pattern, and an extreme vertical jaw gape, the evidence moves far beyond the realm of ambiguous wildlife sightings. It presents science with a cohesive, living architectural profile that perfectly matches the historical baseline of Thylacinus cynocephalus, forcing modern zoology to re-examine the absolute boundaries of extinction. If such a distinctive predator is truly navigating the wilderness with these precise anatomical structures, how has forensic science managed to overlook its physical presence for nearly a century? The 1936 baseline and the history of the bounty. To fully comprehend why modern science demands such an absolute standard of physical proof, one must look back at the exact historical baseline established when the species officially crossed the threshold into extinction. The thylacine did not naturally fade away because of a changing climate or an evolutionary dead end, nor did it lose out in a fair fight against nature. Its disappearance from the Australian ecosystem was a calculated, state-sponsored, and systematically executed eradication program. We did not merely lose the Tasmanian tiger, we actively hunted it until there was nothing left but museum specimens. And that realization leaves a permanent, bitter mark across the history of modern conservation biology. Long before European settlers stepped foot onto the rugged shores of Tasmania, the thylacine was the undisputed apex predator of the island. It stood as a beautiful testament to a biological concept called convergent evolution, which is the process where two unrelated organisms develop nearly identical physical traits because they occupy the exact same ecological niche. Even though the thylacine was a strict marsupial that raised its tiny, undeveloped young inside a backward-facing belly pouch, it looked hunted, and functioned almost exactly like a canine or a wolf. For thousands of years, it kept the island's ecosystems perfectly balanced, preying primarily on native wallabies, kangaroos, and smaller ground-dwelling marsupials. Everything changed in the 19th century when European colonists arrived with thousands of sheep, looking to turn the pristine island into a massive, lucrative wool-producing empire. It did not take long for these settlers to notice that their livestock was occasionally turning up dead. Rather than looking closely at their own poorly managed farming practices or blaming the packs of feral domestic dogs that they themselves had introduced, the farmers looked out into the dark, shadowy tree line of the bush and found their perfect scapegoat. They branded the thylacine as a bloodthirsty, ruthless monster, a demonic sheep-killing tiger that would ruin the colony's economy if it was not completely wiped out. The media and agricultural reports of the era whipped the public into a total frenzy, publishing wildly exaggerated, fabricated stories of tigers raiding homesteads and slaughtering entire flocks of sheep in a single night just to drink their blood. By 1830, private land companies were offering cash rewards for every dead tiger brought into their offices. Then, in 1888, the Tasmanian government officially stepped in and established a formal, state-funded bounty system. They placed a price of £1 per adult tiger and 10 shillings per pup. In the economy of the late 1800s, that was a massive amount of money for a poor bushman, laborer, or farmer. The slaughter that followed was absolute chaos. For over 20 years, the wilderness was flooded with professional bounty hunters, traps, strychnine poison, and specialized hunting hounds. Between 1888 and 1909, the government officially paid out thousands of bounties, but the actual number of slaughtered animals was likely double or triple that amount, as many killed tigers were never formally registered or were lost in the dense brush. The animals were shot on sight, poisoned at watering holes, and driven completely out of their native hunting grounds. By the time the government finally woke up and canceled the bounty system in 1909, it was already too late. The population had been completely shattered, pushed far past the point of genetic recovery. What followed was a heartbreaking, slow-motion disaster. By the 20s, sightings of the tiger in the wild had become incredibly rare. Occasionally, a stray, starving animal would be shot by a confused farmer, but the vast forests had grown completely quiet. Yet, despite the obvious, flashing warning signs that the species was on the absolute brink of total annihilation, the government and the public remained completely indifferent. The tragedy reached its ultimate, symbolic climax on September 7, 1936, at the Hobart Zoo. Living in a small, barren, concrete and wire enclosure was a single thylacine named Benjamin. He was the very last known member of his entire species. On a freezing Tasmanian night, the zookeepers, out of pure, lazy neglect, locked Benjamin out of his sheltered sleeping quarters, leaving him completely exposed to the elements. The animal was forced to endure blistering nighttime temperatures on the cold concrete without any protection. By the time the sun came up the next morning, Benjamin was dead. He had died of hypothermia. In a twist of cruel, dark irony, the Tasmanian government had officially passed a strict law to grant the thylacine full environmental protection just 59 days before Benjamin died. They finally decided to save the tiger right when there was no longer a tiger left to save. The body of the last known apex predator on the island was casually thrown into a dumpster, and the zoo did not even bother to take a formal photograph to mark its passing. The species was gone, erased in less than a single human lifetime by pure ignorance and cruelty. But if the very last tiger died in a concrete cage in Hobart, why do so many modern sightings pop up hundreds of miles away on the Australian mainland? The geographic anomaly, mainland frontiers. To make this entire mystery even more mind-boggling, you have to look at where a huge percentage of modern, compelling sightings and remote trail cam recordings are actually coming from. Logically, if the Tasmanian tiger managed to survive its state-sponsored eradication, It should be hiding in the deepest, darkest, most impenetrable corners of Tasmania. It should be roaming places like the dense, wet wilderness of the Tarkine rainforest or the rugged Southwest National Park. These are places so dense, thick, and unforgiving that human beings rarely set foot there, making them perfect sanctuaries for a remnant population trying to evade human contact. But that is not what is happening. Instead, a massive wave of the most convincing modern footprints, eyewitness accounts, and night vision recordings is popping up hundreds of miles away, completely across the ocean on mainland Australia, specifically in the thick, rugged bushlands of Victoria and South Australia. This creates an absolutely massive geographic and evolutionary paradox that drives mainstream scientists completely insane. According to the firmly established fossil record and archaeological data, the thylacine went completely extinct on mainland Australia roughly 3,000 years ago. It vanished from the mainland long before European settlers ever arrived with their guns, traps, and sheep bounties. Fossilized remains found in caves across Western Australia and South Australia show that the animal once dominated the entire continent, but then it abruptly vanished from the mainland timeline, leaving the island of Tasmania as its final isolated stronghold. The scientific consensus points squarely at two major factors to explain this ancient mainland extinction: a shifting, drying climate that altered their hunting grounds and the introduction of the dingo, Canis lupus dingo. Dingoes were brought to the Australian mainland by seafaring humans thousands of years ago, and they immediately filled the exact same ecological niche as the thylacine. Because dingoes are highly intelligent, highly social pack hunters, they easily outcompeted the solitary, slower-moving thylacine for food and resources, driving them to extinction across the entire mainland continent. However, because dingoes never managed to cross the treacherous waters of Bass Strait to reach the island of Tasmania, the isolated island became a safe, pristine sanctuary where the thylacine could survive and thrive for thousands of more years without facing canine competition. So, if the mainland population was completely wiped out millennia ago, how on earth can researchers claim to be filming them roaming around the forests of Victoria today? It seems completely impossible that an animal could magically reappear on a landmass where it has been dead for 3,000 years. Cryptozoologists, independent trackers, and locals have floated several wild, fascinating theories to explain this geographic puzzle. The most popular theory is that the tiger didn't naturally survive on the mainland. Instead, it was secretly or accidentally put back there by humans during the 19th and 20th centuries. Back during the height of the Tasmanian bounty era, it was highly common for live thylacines to be captured by private collectors, traveling circuses, and exotic animal showmen. These handlers frequently traveled across the mainland states, showing off the bizarre, striped marsupial beasts to curious crowds in Melbourne, Sydney, and rural farming towns. Local folklore across the state of Victoria suggests that multiple instances occurred where these captive tigers managed to escape their cages, fleeing directly into the dense, vast eucalyptus forests of the Otway Ranges or the Wilson's Promontory. Furthermore, rumors have long circulated among historical researchers that several wealthy, sympathetic landowners in the early 1900s, realizing that the tiger was facing total annihilation in Tasmania due to the government bounty, secretly bought breeding pairs from Tasmanian trappers and deliberately released them into the wild Australian bush on the mainland to act as a secret insurance population. While that sounds like a thrilling, romantic narrative, mainstream biologists find the theory incredibly hard to swallow. For an animal population to survive for nearly a century, you need more than just one or two lucky escapees. You need a viable, healthy, and genetically diverse breeding population. If a large, striped, carnivorous marsupial has been actively breeding, hunting, and dying in the heavily populated and thoroughly explored state of Victoria for the last 100 years, it seems mathematically impossible that not a single one has been hit by a car, shot by a deer hunter, or found dead by a hiker. The bush is vast, Yes, but human encroachment on the mainland is massive. Yet, the reports continue to flood in. Field naturalists frequently document plaster casts of footprints that show the characteristic asymmetrical four-toe arrangement and the distinctive heavy heel pad of a marsupial carnivore rather than a wild dog. Locals in small, rural timber towns swear they have seen the telltale stripes, the stiff tail, and the awkward hopping gait cutting across quiet country roads in the dead of night. Whether these mainland sightings are a testament to the animal's incredible, ghostly ability to evade human detection against all mathematical odds, or simply a case of mass psychological projection fueled by a collective national desire to see a legendary icon return, the geographic paradox remains one of the most frustrating, unexplained chapters of the entire thylacine mythos. But what if we don't have to rely on hidden cameras in the woods to bring this ghost back to life? The laboratory resurrection, rewriting the code. If all those remote trail cam videos and night vision recordings eventually turn out to be nothing more than natural misidentifications or optical illusions, it might not actually matter in the long run. We may not need to find a hidden, miraculous breeding population of Tasmanian tigers hiding out in the deep, forgotten rainforests of the Tarkine to see one alive again. While field researchers are busy combing the dense bush for tracks and hairs, an entirely different kind of tracking is taking place inside some of the most advanced genetic engineering laboratories on the planet. Science has stepped directly into the territory of biological synthesis, and researchers are actively working to play God by dragging the thylacine back from the dead, one single line of genetic code at a time. This is no longer a loose, hypothetical plan for the distant future. It has transformed into an incredibly well-funded, fast-moving reality. At the absolute forefront of this scientific frontier is a massive biotechnology company called Colossal Biosciences, which operates a multi-million-dollar genetic engineering crusade. Partnering directly with the University of Melbourne's TIGAR Lab, the Thylacine Integrated Genomic Restoration Research Lab, led by world-renowned geneticist Dr. Andrew Pask, these institutions have transformed de-extinction from a wild fantasy into a legitimate, heavily backed branch of modern conservation biology. The process they are using to resurrect the Tasmanian tiger is an absolute marvel of molecular paleontology, and it relies heavily on revolutionary CRISPR gene editing technology. To bring back an extinct animal, you first need a highly accurate blueprint of its genetic material. For a long time, scientists thought this was impossible because DNA degrades rapidly after an organism dies, leaving behind nothing but broken, unreadable fragments. But in a massive scientific breakthrough, Dr. Pask's team managed to extract high-quality, ancient DNA from a 108-year-old thylacine pup that had been safely preserved in a jar of alcohol at the Victoria Museum. Because the alcohol kept the genetic strands intact, scientists were able to sequence what is now widely considered the highest quality, most complete platinum-level genome of any extinct species on the planet. However, having a complete blueprint isn't enough. You cannot just inject raw, ancient DNA into a petri dish and watch a tiger grow. You need a living canvas to write that blueprint onto. To do this, scientists selected the thylacine's closest living marsupial relative, a tiny, mouse-sized creature called the fat-tailed dunnart, Sminthopsis crassicaudata. Even though a dunnart looks nothing like a wolf-sized apex predator, their DNA sequences are incredibly similar under the microscope, sharing a vast majority of their evolutionary code. The scientists take living stem cells from the fat-tailed dunnart and use CRISPR as a pair of hyper-precise, microscopic biological scissors. They systematically cut out the specific sections of the dunnart's DNA that make it a small, insect-eating mouse and replace them with the resurrected synthetic genes of the thylacine. They are meticulously altering the code to command the cell to develop the physical traits of the tiger, the 15 to 20 dark lumbar stripes, the massive vertical jaw gape, the rigid marsupial tail, and the specialized carnivorous digestive system. Once this hybrid, edited cell line is perfected, the next massive hurdle is turning that microscopic cell into a living, breathing animal. Because marsupials give birth to tiny, highly undeveloped embryos that finish growing inside an external belly pouch, the reproductive process is vastly different from placental mammals. The TIGRR lab has successfully pioneered advanced assisted reproductive technologies, ART, including marsupial IVF and artificial womb technologies, specifically designed to host and grow these genetically engineered thylacine embryos. The ultimate goal of this massive genetic project isn't to create a lonely, freakish science experiment to show off inside a paid zoo enclosure. The scientists at Colossal and the University of Melbourne argue that bringing back the thylacine is an act of crucial ecological restoration. When humans wiped out the island's apex predator, it triggered a devastating biological collapse known as trophic downgrading. Without a top-tier hunter to cull the weak and the sick, native herbivore populations ran rampant, overgrazing the landscape, and diseases began spreading uncontrollably through native mammal population. Biologists believe that reintroducing a functionally identical Tasmanian tiger back into the wild could completely stabilize the fragile Tasmanian ecosystem, helping to restore balance to the forests. But as technology races forward, it forces us to confront some deeply unsettling ethical questions. If we successfully resurrect an animal that has been gone for a century, how will it learn to survive? Will a creature raised entirely inside a sterile plastic lab or a synthetic incubator understand how to hunt wild wallabies? Or will its natural, prehistoric instincts seamlessly kick in the moment its paws touch the dirt? Are we truly fixing a tragic historical mistake? Or are we just letting our human arrogance create a genetic copy of a ghost? So, where does that leave us right now with the mystery of the Tasmanian tiger? The proxy species dilemma and the illusion of de-extinction. If the laboratories successfully complete their genetic editing sequences and bring an embryo to full term, science will confront an entirely new, deeply unsettling philosophical and biological crisis: the definition of a true species. In the public eye, replacing parts of a living creature's genetic code using modern technology implies a flawless resurrection. The common assumption is that if an animal looks like a thylacine, possesses its dark lumbar stripes, and displays its distinctive wide jaw gape, then the thylacine has officially returned to our world. However, within the rigorous fields of evolutionary biology and taxonomy, a growing number of researchers are raising warnings that this laboratory-created organism will not actually be a Tasmanian tiger at all. Instead, it will be a highly engineered proxy, a modified fat-tailed dunnart masquerading in the physical shell of a ghost. This distinction moves far beyond simple semantics. It hits at the core of how a species is formed over millions of years of natural selection. An animal is not merely the sum of its raw DNA sequence written into a computer database. A species is an incredibly complex, living tapestry woven from its genetics, its maternal environment, its microbiome, and its generational learned behaviors. When a native thylacine gave birth in the wild a century ago, the tiny, blind embryo crawled into its mother's backward-facing pouch. Inside that highly specialized environment, the young pup did not just consume milk. It ingested a specific cocktail of maternal antibodies and essential gut microbes unique to the Thylacinidae family. These microscopic organisms calibrated the pup's immune system, regulated its metabolism, and dictated how its digestive tract would break down the raw meat of wild prey. A synthetic thylacine embryo growing inside an artificial womb membrane or an engineered surrogate host in a sterile lab will completely lack this evolutionary microenvironment. It will be born into a biological vacuum. Lacking the generational line of maternal pouch biology, its immune system and internal microbiome will be entirely artificial, leaving it fundamentally altered from the historical baseline of the animals that hunted the Tasmanian wilderness prior to 1936. Furthermore, we must confront the total absence of behavioral transmission. In the wild, apex predators are not born with their entire hunting strategy pre-programmed into their brains like a piece of software. While basic predatory instincts are hardwired, the hyper-specific, fine-tuned behaviors required to navigate an ecosystem are passed down from parent to offspring through active training and observation. A wild thylacine learned from its mother how to stalk a wallaby through the dense ferns of the Tarkine, how to utilize its rigid tail as a precise kinetic counterweight during high-speed bounding turns, and how to read the complex olfactory map of the Australian bush. The laboratory-born proxy will have no mother to teach it these survival skills. Raised by human technicians behind glass partitions and nourished by automated delivery systems, its psychological development will be a total anomaly. When this animal is eventually released into the wild to fulfill its intended ecological role as an apex hunter, it will step onto the dirt as an behavioral orphan. Biologists openly wonder if such an animal will even know how to survive, or if its hardwired predatory instincts will misfire, leaving it unable to accurately read its environment or successfully hunt wild prey. By introducing a synthetic proxy predator into a fragile modern ecosystem, science may inadvertently introduce a chaotic wild card rather than a healing balance. If the engineered animal behaves differently than the historical thylacine, if its altered gut microbiome changes its dietary preferences, or if its lack of generational training causes it to target easy domestic livestock rather than elusive native game, the entire restoration project could collapse into an environmental disaster. It raises a haunting question that no sequence of gene editing code can answer. In our desperate, guilt-ridden attempt to undo the slaughter of the past, are we actually restoring a beautiful piece of lost nature, or are we simply creating a beautiful, highly advanced illusion of life? The true status of the thylacine remains balanced on a razor's edge between the ancient wilderness and modern technology. Whether the animal is actively hiding in the deep, unmapped fern gullies of the Tarkine rainforest or waiting to be born inside a sterile laboratory petri dish, one thing is certain: the apex predator has never truly left our consciousness. Humanity remains permanently haunted by the ghost of 1936, caught between the desperate hope that nature saved the tiger from our greed and the terrifying reality that science is about to build a new one. Whether the thylacine is ultimately found in the shadow of a rainforest fern or synthesized under a laboratory lens, it forces us to confront our deepest ecological responsibilities. We remain permanently haunted by the ghost of 1936. If you want to track more biological mysteries, subscribe for future deep dives.